Hi guys, in today's video I will be discussing with you about oral cancer. It is considered as one of the toughest topic while preparation, but it is one of my favorite topics, thus I will be discussing with this topic. While my preparation, my study plan was to combine two subjects, one basic science subject and one clinical subject. So all my videos will be combining two subjects, hitting two goals at one time. And in this video, I will be covering topics related to pathology that are applied in oral surgery. So now about oral cancer. Oral cancer is considered worldwide as sixth most common cancer, while in India it is considered as third most common cancer. As oral cancer is a very vast topic, thus I will be covering in 3 to 4 videos, and I will be touching upon all basic points that are asked at entrance exam level and your BDS exam level. So the topics that will be covered in this video are etiology of oral cancer, cell cycle and checkpoints, cell injury, molecular basis of oral cancer, apoptosis, checkpoints, oncogenes and tumor suppressor genes. So these are the most frequently asked questions, either in short answer form or long answer form. So kindly make note of it. And guys, stay tuned till the end of this video as lots of MCQs are coming up, and try commenting answers in comment section, and I will be discussing in my next video. So I will be describing this tough looking process in a very simple way, as it is important to understand the whole process behind each and everything. Only then you can retain retain MCQs till the end of the exam and solve all related MCQs. So first we will be discussing about etiology of the oral cancer. You have to remember basically 4 or 5 main etiological factors of oral cancer. First is tobacco consumption, second is alcohol, third is areca nut or betel nut, fourth, sharp teeth which are acting as irritant to the adjacent mucosa or ill-fitting dentures. Then unhealthy diets, low intake of fruits, vegetables, viruses. This is important question again. Human papilloma virus, HPV 16 and 18. These are frequently asked MCQ questions. And last is oral candidiasis. Mainly you have to remember all these etiology. So here in this slide we will be discussing mainly about areca nut. What does areca nut do to the oral mucosa? First, it causes release of oxygen-free radicals, and because of this oxygen-free radicals, it causes inflammation of the surrounding mucosa by various factors like COX, interleukins, etc. And thus, because of this redox-related inflammation, there is precancerous malignant transformation. Second, it causes genotoxicity, cytotoxicity, autophagy, and angiogenesis, and all these factors lead to malignant transformation. Third, it calls EMT. Now, what is EMT? EMT is epithelial mesenchymal transition. So, epithelial cells gets the ability to transit into mesenchymal cell, and when it converts into mesenchymal cell, it gets the capability for migratory capacity basically. So, because of this migratory capacity, it leads to metastasis. Lastly, areca nut also facilitate chemoradiosistance and cancer stemness. Now, what is cancer stemness? Means they are able to convert these transformed cells into their progeny. Thus, all these factors lead to cancer progression. Now, we will be discussing about cell cycle and its checkpoints. So, what happens for cell cycle to proceed? It has to pass through certain checkpoints or transition points. And if DNA is damaged due to any reason, these checkpoints will get activated and cell cycle won't progress further. It will be arrested at that particular stage only. Or in some cases, cell will only divide when cell will receive certain stimuli like hormones, cytokines, etc. Now we will be studying about CDKs. What are CDKs? So in simple terms, CDKs are cyclin dependent kinase, or kinase are enzymes. So kinase which are dependent on cyclin. Remember cyclin for cycle. So there are various cyclin dependent CDKs that are acting on this cell cycle. So first we will be studying about cyclins that are acting on G1 estase or mid to late G1 phase. So what cyclins are acting at this phase are cyclin D or CDK4. So what happens when these are activated? They makes the cell enter into mitosis. Secondly, checkpoints that are acting on G2 phase are cyclin A, CDK1. Now we will be talking about inhibitors for this cell cycle. Inhibitors, mainly you have to remember this P21 inhibitor. If it is activated, then cell won't undergo into mitosis. And mainly among all these cyclins, you have to remember cyclin D, which is most commonly involved in all human cancers. Okay. Now we will be discussing about tumor suppressor genes and oncogenes. So what are oncogenes? Oncogenes are genes that induce transformed phenotype. Transformed phenotype means altered cells. So when they are expressed in a particular cell by promoting increase in cell growth. So when they are expressed, they cause increase in cell growth. And cancer cells generally develop for gain of function mutation. Remember, gain of function mutation. So because of this mutation, the proto-oncogene, which is normal gene, normal cellular gene is proto-oncogene, and if there is mutation, it will convert into oncogene. So most commonly oncogene that you have to remember that is involved in all human cancer. Approximately 30% of the all human tumor is RAS gene. RAS gene. So what does RAS gene do? RAS gene flips back and forth from its inactive form to its active form. This is its inactive form. This is the RAS gene. It will flip back and forth from its inactive to active form. How does it flip form? By converting GDP molecule into GTP. By addition of one phosphate molecule, it will convert into its active form. And by activating various molecular pathways, it will cause release of MYC protein, and this MYC protein causes cell cycle to progress. So what will happen if there is point mutation in this RAS gene? So RAS gene will be trapped in its activated form, that is the phosphorylated form, and the cell will continue to generate this MYC protein, and there will be continuous progression of this cell cycle. So next genes that are involved in carcinogenesis is tumor suppressor genes. So what are tumor suppressor genes? Genes that are normally prevent uncontrolled growth of the cell, but when they are mutated, when the tumor suppressor gene is mutated or lost from a particular cell, they allow the transformed phenotype. What is transformed phenotype? That malignant cells to develop its progeny. So most commonly mutated tumor suppressor gene you have to remember is P53. So which is also monitor for internal stress of the cell. So cells that have damaged damaged DNA, if the P53 is normal, not mutated, so it by acting at various pathways through various pathways, it will cause either to undergo cell into a senescence stage, or it will cause apoptosis of the cell. But when this P53 is mutated or lost, or because of any ionizing radiation, carcinogen, mutagen, this cell that has damaged DNA along with P53 mutation, there will be no cycle arrest. The cell won't undergo into apoptosis. The cell won't undergo into senescence, and there will be expansion and additional mutation, and the cell will undergo into a malignant formation of malignant tumor by generating its progeny. Okay. Now another tumor suppressor gene that you have to remember is retinoblastoma gene. So retinoblastoma gene will act at G1S checkpoint. Cell cycle we have discussed previously. At G1 to S transition phase, there will be the gene that will be acting is retinoblastoma. So growth inhibitors like tumor growth factor beta P53, what it will do? It will stimulate CDK inhibitors. This CDK inhibitors inactivate cyclin D, cyclin E. So this retinoblastoma gene will stay in hypophosphorylated state, means phosphate won't attach to retinoblastoma gene, and this E2F is not released, and further it cannot give signals to DNA for further transcription. Now what will happen if there are growth factors like EGF, PDGF? It will activate. Here the growth inhibitors were inactivating cyclins, but when growth factors are present, they are activating cyclins like cyclin D, cyclin E, and when it is activated, it will cause phosphorylation added in retinoblastoma gene. And when the phosphorus are added, it will release E2F. And when this E2F molecule is released, it will send signal to DNA. And then there will be transcriptional activation and cell will divide. Cell will undergo to mitosis and further progression of tumor when this gene is mutated. Now, third you have to remember is APC protein. Now, what does APC do? It forms a macromolecular or destruction complex with beta-catenin. So, beta-catenin, when it forms a macromolecular complex, it causes lysis of beta-catenin and it won't send signals to DNA for further transcription and there will be no proliferation. But when there is growth stimulation, WMT stimulation, or when the gene is mutated, what will happen? This complex, destruction complex won't be formed and beta-catenin is free to send signal to the DNA for further proliferation. So in both these cases, there will be proliferation and cell will undergo even mitosis and tumor will form. Now we will be studying about apoptosis. So apoptosis for molecule, if we have to go into the molecular level, it will involve two pathways, extrinsic and intrinsic pathway, and both these extrinsic and intrinsic pathways will lead to final common pathway. So what happens in extrinsic pathway? Some extrinsic molecules like Fas ligands. So what are Fas ligands? Some extrinsic ligands are binding to the cell which are activating cellular machinery so that cell will undergo into apoptosis. You have to remember mainly Fas ligand. Now for intrinsic pathway, it is activated only when DNA is damaged. So because of reactive oxygen species or some chemicals, radiation, if the cell DNA is damaged, what will happen? It will, by molecules like BAX and BAX, it will cause increase in permeability of mitochondria. And once permeability of mitochondria is increased, it will cause leakage of cytochrome C. So when cytochrome C is leaked into the cytoplasm, it will cause activation of caspases 9. So caspases 9 will activate caspases 3, which is the execution cascade for apoptosis, and then cell will undergo into apoptosis state. But when there is mutation of P53 gene, it will cause inactivation of BAX and thus this whole process is inhibited and cell won't undergo into apoptosis. Secondly, if there is increase in anti-apoptotic factors like BCL2, BCLXL, the transition of the cell is shifted into anti-apoptotic site and cell won't undergo into apoptosis. Lastly, when there is release of IAPAs. What is IAPAs? It is inhibitors of apoptotic proteins. So it will inhibit both capsid 9, and once capsid 9 is inhibited, there is no executional capsid 3, and cell won't undergo into apoptosis. So basically, this is in short related to what are the pathways for apoptosis. Now we will be discussing about MCQs. The answers of all these questions I will be discussing in my next video. So first MCQ is which of the following oral cancer subtype has a worse prognosis? Either buccal mucosa, hard palate, oral tongue, or second, dissection of cancers of more than 1/3 of the oral tongue are best managed by either primary closure, grafting, alloderm graft, skin graft, radioforum flap, or extension. So third question is which subside of the oral cavity has the highest frequency of oral cancer? Buccal mucosa, hard palate, oral tongue. Fourth question is which of the following is not a risk factor for oral cancer? Tobacco, alcohol, fanconi anemia, herpes simplex virus, or HIV infection. Fifth and the last question is the pre-malignant lesion that is the highest risk of malignant transformation. Either homogeneous leukoplakia, erythroplakia, non-homogeneous leukoplakia, oral lichen planus, or oral submucous fibrosis. So guys, these are all my references and I suggest you to go at least a chapter on neoplasia from Robbins. Other two books you won't require at your level. So guys, that's all for today's video and I hope this video is informative for you all. and meet you next time with continuation of this video and some other interesting topics. Till then, like, share, subscribe my channel and study hard for exams. Bye bye.